Here we will understand the structure of DNA. So, how is this biomolecule structure and what are its constituent molecules? So, let's find out in this lecture. The structure of DNA molecule was discovered in the earlier years of 1950s. Note that the structure was discovered, not the molecule. The discovery of DNA molecules predict to the 18th century, but the DNA was ignored for long time and it was protein that grabbed a lot of attention. However, post Griffith transformation experiment and other genetist work, it shifted the focus towards the DNA. Francis Crick and James Watson, two scientists in the field of genetics, they work on the structure of DNA. They even got Nobel Prize for the discovery of double helical structure of DNA. So let's understand how exactly this double helical structure of DNA looks like. We may have come across the structure of DNA somewhere. Yes, according to Watson and Crick, the DNA is made up of three major components. First is pentose sugar molecule called as deoxyribose. Yes, deoxyribose. How is this obtained? It is obtained by removing one oxygen atom from a ribose molecule. If one oxygen atom removed from the ribose, it forms deoxyribose. So thus the name deoxyribonucleic acid. Now see the second component. Second is phosphate. This helps bind the two deoxyribose molecule together. Now the third and the fifth carbon atom of the adjacent deoxyribose molecule are bound by the phosphate groups. See the third component. Lastly, the nitrogenous base forms the core of the double helix. Yes, so these are the four different molecules that bind in pair. Just see, these are the four different molecules that binds in pair. Which are these here? Guanine and adenine, cytosine and thymine. So two of them are called as purine base, whereas the other two are called as pyrimidine base. So guanine and adenine are purine bases and cytosine and thymine are pyrimidine bases. So can any purine bind with any pyrimidine? Answer is no. Adenine always bind. A for adenine. So adenine always bind with thymine. Yes, while guanine always bind with cytosine only. So these molecules are bound with each other with the help of hydrogen bonds. You can see in between the adenine and thymine, here is the hydrogen bond. This point what I am putting, that is the hydrogen bond. So adenine binds with thymine with the two hydrogen bond. You can see here two hydrogen bond. Whereas guanine and cytosine, however, bind with the three hydrogen bonds. Now, did you know that one unit consisting of deoxyribose molecule, one nitrogenous base and a phosphate group is called as nucleotide? Let's see here. You can see in this diagram, that here one unit it consists of deoxyribose. Yes, you can see here deoxyribose sugar, deoxyribose, one nitrogenous base and a phosphate group. So that is, this one unit is what called as nucleotide. So what is nucleotide here? Nucleotide is deoxyribose sugar, nitrogenous base and phosphate. This is nucleotide. And what if the phosphate group is removed from this nucleotide unit? See here in the nucleotide, here phosphate group is there. So what if this phosphate group is removed? Then it is called as nucleoside then. Yes. So nucleoside is deoxyribose sugar plus nitrogenous bases. Now, is there any way measuring a DNA molecule? You can see here in this slide. So what about its dimension? What about its dimension? Let's see here in this slide. The dimension of DNA are very simple and easy to remember. The diameter of double helix is around 2 meter. You can see here 2 nanometer. Yes. And the double helical strand has one major and one minor grooves. Yes, one major groove and one minor grooves. So that make up one turn. So hence here we'll see the length of the one turn, that is around 3.4 nanometer. Yes, 3.4 nanometer, and there are around 10 base pairs in one turn. You can see here one turn. This is the complete one turn, and here 10 base pairs in one turn. 1 to 10 is mentioned here. Now, can you help me with the distance between the two adjacent base pair? The distance is, how much is the distance? That is 0.34 nanometer. Yes. So it's simply how is this distance between the two adjacent base pair? Yes, in between the two adjacent base pair is 0.34. It's very simple. How? You can see 3.4 divided by 10. Yes. So 3.4 is what? The length of the one turn, and divide by the 10 base pairs. Then we will get 0.34. Yes. So this is how the molecules are arranged. So in this double helix, let's see here. Yes. So in this double helix, here the backbone is made up of deoxyribose and phosphate groups. Yes, you can see here deoxyribose and phosphate group. Yes, whereas the portion that forms the core is made up of nitrogenous bases. Yes, this core part, the middle part. So this is made up of nitrogenous bases. Now, can you guess the chemical nature of DNA? Since it contains nitrogenous bases, you would say that DNA is basic in nature. Yes, you may say that it is basic in nature as nitrogenous bases are present, but that's not true. DNA in fact is acidic in nature. The presence of phosphate group makes the molecule acidic in nature. Presence of this phosphate group makes the molecule acidic in nature. So what about its physical appearance? Yes, can you tell me how long this double helix be in length? So let me tell you a very interesting fact. If all the DNA, yes, all the DNA from a single cell is pulled out, yes, and stretched, then it would be touch approximately 2 meters length. Yes, that's long the DNA is. But if the DNA is stretched so long, then how does it fit in such a tiny nucleus? We know that the cell is extremely tiny and the nucleus occupy a very small portion of the tiny cell. So how does this long stretched DNA does fits in such a tiny space? The answer is quite simple. How do we fit a telephone wire in the small space? Yes, so that's using the coiling process. So similarly, DNA also gets fit in the tiny nucleus. So that we will discuss in the next part, that is DNA packaging. Here I want to show you one animation, 3D animation about the structure of DNA. So let's see here. You can see here DNA, double helical structure of DNA is a polymer of two strands of nucleotides, yes, which are hydrogen bonded to each other to form double helix. Nucleotides are molecules that consist of deoxyribose sugar, yes, deoxyribose sugar, a phosphate, and one of four nitrogenous bases, the phosphodiester backbones that consist of alternating sugar and phosphate groups. The nitrogenous bases includes cytosine, thymine, adenine, and guanine. Cytosine forms here 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bond with adenine. So this is referred to as complementary base pairing. The double helix will have one strand oriented in 5:3 direction and hydroxyl group of the deoxyribose sugar, and the other strand oriented to 3:5 direction. This shows the anti-parallel nature of the DNA strand. The complementary base pairing in the structure of DNA that allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template that creates. Double-stranded DNA is a polymer of two strands of nucleotides which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of four nitrogenous bases. The phosphodiester backbone consists of alternating sugar and phosphate group. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms here 3 hydrogen bond with guanine, and thymine forms 2 hydrogen bonds with adenine. This refers to the complementary base pairing, and the double helix will have one strand oriented into 5:3 direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in 3:5 direction. Yes, this shows the anti-parallel nature of the DNA strand and the complementary base pairing in the structure of DNA that allows the replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template strand for the creation of new double strand. So in this way, in this animated video, we had seen the structure of DNA in detail and about its major component. Now in next lecture, we will discuss in detail about DNA packaging in prokaryotes and in eukaryotes.